Basic principles of immunologic diseases in the skin. The skin is much more than a protective covering. It acts as a physical barrier that helps maintain the body's homeostasis while also serving as an active part of the immune system. Under normal conditions, the skin mounts carefully controlled immune responses against harmful microorganisms and foreign substances. These responses are strong enough to eliminate threats, but are also limited to prevent damage to healthy tissues. When these immune mechanisms become dysregulated, the immune system may attack harmless substances or even the body's own tissues, resulting in immunologic and inflammatory skin diseases. One common example is allergic contact dermatitis, or ACD. This condition develops after repeated exposure to allergens such as chemicals, metals, or plants like poison ivy. These substances, known as haptens, combine with skin proteins to form antigens that trigger an immune response. During the first exposure, immune cells called dendritic cells activate T lymphocytes and create memory T cells. Upon later exposure to the same allergen, these memory T cells rapidly trigger inflammation, causing redness, swelling, itching, and eczema. Regulatory T cells help limit this response, but when regulation fails, chronic dermatitis may occur. Drug reactions. These are life-threatening skin conditions caused by abnormal immune responses to medications. Early recognition is very important because the first step in treatment is the immediate discontinuation of the offending drug. One example is Stevens-Johnson syndrome, or SJS. In this condition, CD8-positive T cells attack keratinocytes, leading to widespread skin and mucosal damage. Certain HLA genetic types, especially in Asian populations, increase the risk of developing this reaction. Another severe reaction is DRESS syndrome, which stands for drug reaction with eosinophilia and systemic symptoms. It is characterized by eosinophilia, lymphadenopathy, fever, and involvement of internal organs. It may also cause herpes virus reactivation and can lead to long-term autoimmune complications. A less severe but characteristic reaction is the fixed drug eruption. In this condition, skin lesions repeatedly appear at the same location whenever the patient takes the same medication. This happens because resident memory T cells remain in the previously affected skin and quickly react upon re-exposure. There are three main mechanisms that explain immune-mediated drug hypersensitivity. Hapten model: the drug binds to carrier proteins, forming immunogenic complexes that activate the immune system. Altered peptide model: the drug binds directly to HLA molecules, changing the peptides presented to T cells. PI concept, or pharmacologic interaction with immune receptors: the drug directly interacts with the T cell receptor or HLA without requiring antigen processing, leading to rapid T cell activation. Another important condition is atopic dermatitis, also known as eczema. It is characterized by dry, itchy, inflamed skin and commonly begins during childhood. Many patients later develop asthma, allergic rhinitis, or food allergies, a progression known as the atopic march. Research has shown that mutations in the filaggrin gene, which is essential for maintaining the skin barrier, contribute significantly to disease development. Atopic dermatitis also involves abnormal immune responses, particularly type 2 immunity with increased IgA, IL-4, and IL-13. In addition, changes in the skin microbiome, especially overgrowth of Staphylococcus aureus, worsen inflammation. New biologic therapies such as dupilumab, which blocks IL-4 and IL-13 signaling, and JAK inhibitors have greatly improved treatment outcomes for many patients. Psoriasis. A chronic systemic inflammatory disease. Although it is commonly recognized as a skin disorder, recent research has shown that psoriasis affects much more than the skin. It is associated with increased risks of cardiovascular disease and can even reduce life expectancy. Historically, psoriasis was believed to be simply a disorder of excessive keratinocyte proliferation. However, this understanding changed when researchers discovered that the immune system, particularly T cells, plays a central role in the disease. The effectiveness of treatments such as cyclosporin and lymphocyte-depleting agents provided strong evidence for this immune involvement. The next major breakthrough was the development of anti-TNF biologics, such as infliximab. These therapies significantly improved disease management and transformed the treatment of moderate to severe psoriasis. Our current understanding focuses on the IL-23-IL-17 pathway. The Th17 immune response is now recognized as the key driver of inflammation in psoriasis. This has led to the development of highly targeted biologic therapies. For example, ustekinumab blocks IL-12 and IL-23, providing long-term disease control. Secukinumab targets IL-17A and produces rapid clinical improvement. Guselkumab specifically inhibits IL-23 and offers sustained effectiveness with a highly targeted approach. Despite these advances, important questions remain. Researchers are still investigating what triggers the initial activation of the IL-23-IL-17 pathway and whether innate lymphoid cells contribute alongside conventional T cells in disease development. In conclusion, psoriasis is a complex immune-mediated systemic condition. Continued research is expected to improve understanding and lead to even more precise and effective therapies. The next condition is alopecia areata, an autoimmune disease that causes patchy or complete hair loss. In this disease, the immune system attacks hair follicles, especially during the active hair growth phase. Cytotoxic CD8 positive T cells and inflammatory cytokines such as interferon gamma and IL-15 play major roles in damaging hair follicles. Advances in genome-wide association studies, or GWAS, have identified several genes associated with susceptibility to this disease. These discoveries have led to the development of targeted therapies, particularly JAK inhibitors, which have shown encouraging results in restoring hair growth. Another group of disorders is autoimmune blistering diseases, including pemphigus and bullous pemphigoid. These diseases occur when the immune system produces antibodies against proteins that hold skin cells together. In pemphigus, antibodies attack desmoglines, causing fragile blisters and erosions of the skin and mucous membranes. In bullous pemphigoid, antibodies target proteins in the basement membrane, leading to tense blisters beneath the epidermis. Diagnosis is confirmed by immunofluorescence studies and detection of specific autoantibodies. Modern treatments include corticosteroids, immunosuppressive drugs, and the monoclonal antibody rituximab, which removes disease-causing B cells. Researchers are also developing highly targeted therapies such as chimeric autoantigen receptor, or CAAR T-cell therapy, which may provide more precise treatment in the future. Drug hypersensitivity reactions are another example of immune-mediated skin disease. Certain medications can activate T cells abnormally, resulting in severe conditions such as Stevens, Johnson syndrome, and toxic epidermal necrolysis. In these disorders, activated T cells destroy keratinocytes, causing widespread skin damage that can be life-threatening. Overall, recent advances in genetics, immunology, and molecular medicine have transformed our understanding of inflammatory skin diseases. Genome-wide association studies have identified genes that increase disease susceptibility, while biologic therapies have allowed scientists to target specific immune pathways with greater precision and fewer side effects than traditional treatments. In conclusion, immunologic skin diseases result from abnormal immune responses involving genetic susceptibility, environmental triggers, and immune dysregulation. Although much remains to be discovered, research has already produced remarkable advances in diseases such as psoriasis, atopic dermatitis, and alopecia areata. Continued research into immune mechanisms will lead to safer, more effective, and more personalized treatments, improving the quality of life for patients with these chronic skin conditions.